Hello, I'll be presenting to you all today a study done by Kendall titled The Molecular Biology of Memory Storage, a dialogue between genes and synapses. Although this is a wordy and seemingly boring type of study, this study was crucial to the field of neuroscience. In fact, this study alone coined Kendall the father of neuroscience, and he remains a prominent figure in the field today. So what exactly did he accomplish? Kendall wanted to fundamentally understand learning. The way he approached this was through a reductionist approach. Now, you may have heard me use the term reductionism in class in regular discussion, so I wanted to formally introduce what this term means through an analogy. Shown here is a mechanical watch. A mechanical watch can be extremely complex with various levers, gears, pulleys, and springs all working together cohesively to make a functional watch. A reductionist approach would be to look at the most fundamental, simplest form of a watch present and understanding that model to then apply that to more complex models. Just like a complex watch, learning can be extremely complicated as well. Habituation, desensitization, latent learning, and one-time learning are just some of the various forms of learning present. Kendall wanted to explain learning on a molecular level by studying the most fundamental, bare-bones form of learning that he could find. This led Kendall to the Aplasia sea slug. This animal was chosen for its low amount, easy to identify, and easy to stimulate nerve cells. Kendall would stimulate the single nerve cell responsible for a retract mechanism found in the sea slug. This retract mechanism is normally found in nature when the sea slug perceives danger. Kendall observed the biochemical changes found in the sea slug as the slug would slowly learn that the stimuli is no longer harmful and therefore lessening the retraction response. This was accomplished through constant exposure of that specific stimuli. Much like how we stop hearing an annoying buzz in the room or no longer feel the itch of the clothes that we wear, the constant exposure of a stimuli leads us to us ignoring that stimuli. However, there's a specific chain of chemical reactions that lead us to these conclusions. The first step in learning is the release of serotonin. Serotonin can be thought of as the coal fueling the train on its way towards the train track of learning in the presence of a memory. Kendall wanted to study the individual steps of that train track necessary that leads us to learning. He accomplished this by looking at the biochemical changes found, inhibiting each specific biochemical, and then stimulating the sea slug again and observing that response. If the sea slug showed the same weakened response, this meant that that chemical was not necessary for learning, as learning was still present. However, if there was a strong retract mechanism, this meant that no learning had occurred, meaning the inhibited biochemical was crucial and part of this train track. Eventually, Kendall pieced together the train track necessary to go from this first stimuli exposure all the way to learning. This roadmap shows Kendall's discovery. Now, this isn't here to highlight the specific chemicals necessary, but rather to show that a specific train track was found that dictated learning. The crucial chemical that Kendall found was CREB1. CREB1 is responsible for protein synthesis to occur, and protein synthesis is a necessary requirement for long-term memory. If the first parts of the train track can occur, then short-term memory is present, say an exposure, shortly thereafter the same exposure, showing a weakening retract response. Long-term memory, however, was defined as a stimuli exposure, a 24-hour waiting period, and then another exposure. If there was a weakening of that retract response, that means that long-term memory had occurred. Kendall discovered that CREB1 was that crucial defining factor between short-term memory and long-term memory. Thus, a better roadmap of the convoluted idea of learning was deciphered by Kendall. When we forget something we are trying to learn, this is due to failure of CREB1 activation and therefore failure of protein synthesis. This is why we can learn something in the short term and then fail to remember it, say the day after on an exam, in the long-term memory. The only way to remember something long-term is through constant rehearsal and constant exposure to that stimuli. Much like how Kendall constantly exposed that sea slug to that specific retract response, we need to constantly repeat and practice the associations that we make so that we can do better on those reading quizzes. Thank you.